So what we were discussing, that is, it is possibly, we have seen that lead, it is going to mimic the calcium at the site of the Ca2 plus transporter, that is calcium transporters. So how this reabsorption of the calcium which was done by the proximal tubules, we will see. It is going to happen in two-step, that is a two-step process with the calcium channels mediating the transport of this calcium ions across the apical membrane, and as well as calcium ATPase on the basolateral membrane also, which is going to mediate the movement of this calcium ions across the basolateral membrane, and the lead appears to gain access to the proximal tubular cells via the Ca2 plus channels, that is calcium channels on the apical membrane. So the studies in several different cell types they have done, including HEK293 cells, that is nothing but human embryonic kidney cell line, which was demonstrated that lead Pb2 plus can enter the cells via the store operated calcium channels, that is SOCs in short form. So this lead can enter into the cells through the store operated calcium channels. Via these channels, this lead can enter. And it also appears that ORI1 and stromal interacting protein 1, that is STIMT MT1. So which are the critical components of this store operated calcium channels, which plays very important role in the cellular entry of this lead. So data from these particular studies, they have indicated that flux of this lead through the calcium channels is a time and concentration dependent process. It is a time dependent process as well as concentration dependent process, and is going to be approximately 10-fold greater than that which was observed for the calcium Ca. So the exit of this Ca at the basolateral membrane, which involves a calcium ATPase. So the in vivo studies have shown that using this erythrocytes, indicating that lead may substitute for the calcium at the site of this calcium ATPase. The ability of this lead to be transported mainly by the calcium ATPase in the proximal tubular cells, which has been shown since the calcium ATPase is homologous across cell types. So it is very feasible to suggest that this transporter may mediate the movement of the lead across the basolateral membrane of the proximal tubular cells. Understanding? So it can easily mediate the movement of the lead across the basolateral membrane of the proximal tubular cells. So in this way, this store operated calcium channels transporter can work. So this lead may also be transported out of the apical membrane of the proximal tubular cells into the lumen for eventual excretion in the urine. So experimentally, in case of rats, they have done the experiment in case of rats. It suggests that lead can be transported out of the cells as a conjugate. We have already explained in the last class how the other heavy metal ions which are going to be getting conjugated and excreting, right? So conjugate of the glutathione, that is ESH, through this transport of this GSH, lead complex Pb2 plus complex was not demonstrated directly. It is possible that this complex may be transportable form of the lead at the site of this transporter, such as MRP2 or BCRP, which these carriers which are going to be localized in the apical membrane of the proximal tubular cells and mediates the export of this wide variety of these compounds. So in this way, it is very possible that MRP2 and as well as BCRP, which is going to mediate the export of this GSH lead complexes. By the formation of this GSH PB complexes, it is very easy to mediate the export of this complexes through this particular transporter, that is MRP2 and as well as BCRP. In this way, they have they have done one experiment on the rats and they suggested this particular concept. We can easily mediate the export of this GSH PB complexes through these transporter, that is MRP2 and BCRP. Coming for the renal effects of this lead exposure. What are the renal effects? So you all know, owing to this particular role of the kidney in the urinary excretion of the toxicants, so the kidneys, which are going to appear to be one of the primary sites of the accumulation of these toxic compounds, that is in the form of any heavy metal ions, that is in the form of lead, now what we are discussing. So the kidney exposure to the low levels of this lead early in life, which have been shown to lead to glomerular hypertrophy, manifested specifically as an increase in the volume of glomerular capillaries. Exposure to the lead may disrupt glomerular development, of course, which may result in renal insufficiency later in the life. The tubules also gets affected by the exposure to the lead. Acute exposure, which leads to generalized defects in solute and amino acid transport in renal tubules, which leading to the further, we have already discussed this kind of this syndrome, Fanconi syndrome, case of arsenic also, so cadmium also. So chronic exposure to this particular lead may lead to the progressive tubulointerstitial nephritis. Tubulointerstitial nephritis is going to be the further consequence by the exposure to the lead, high level of exposure, that is chronic exposure. So which is mainly characterized by the infiltration of the leukocytes, interstitial fibrosis, and even tubular atrophy. Similarly, when Wistar rats were exposed to this is one kind of rat, Wistar rats, which was exposed to the lead acetate, tubular degeneration has happened. Intraluminal haline cats they have observed, blood vessel congestion, perivascular fibrosis, and vascular edema, and was observed. Since the lead is going to tends to induce the injury in case of proximal tubules, kidney injury molecule, that is 1 KIM. This one I have discussed long back only, that is kidney injury molecule 1, KIM1, and alpha glutathione S transferase, which appears to be the appropriate urinary biomarkers which are used in order to induce, that is in case of this lead for PB induced renal injury, they are using this kind of marker. So one of the primary cellular effects of this exposure of the lead is nothing but the induction of oxidative stress in case of cells of the kidney or the kidney cells. So the exposure of this in the form of the lead acetate in case of mice, which enhanced and observed the enhancement of the production of reactive oxygen species, and it reduced the mRNA expression of the enzymes which is mainly necessary to counteract oxidative stress, that is these are the enzymes, that is catalase, superoxide dismutase, glutathione S transferase, glutathione peroxidase. These are the main enzymes which are involved there in creating oxygen species, that is oxidative stress. So alternatively, the exposure of the mice to the lead, which is also enhanced, which they have observed enhancement of mRNA expression of transforming growth factor also, that is TGF beta 1, and monocyte chemoattractant protein 1, that is MCP1, and even alpha 2 macroglobulin also they observed, which further leads to the inflammatory processes. So the exposure to the lead can also be shown that with respect to the lipid peroxidation and even DNA fragmentation has occurred. So it appears that from these studies, it appears that the mitochondria also plays a very important role in case of lead mediated injury. So the oxidative stress within the cell, which also leads to the alterations in the regulation of this mitochondrial permeability transition pore, that is called as MRTP, which normally helps in mediating the osmotic influx of the water into the mitochondrial matrix. So the exposure of these kidney cells to the lead, which has been shown to induce abnormal opening of this protein, that is pore, that is nothing but mitochondrial permeability transition pore, which further leads to the swelling of the mitochondria, and also you can observe changes in the membrane potential, and initiation of this apoptosis can happen. So exposure of these kidney cells to the lead also leads to structural alterations in the mitochondria, such as distortion of the mitochondrial cristae and swelling and rupture of the outer membrane. So you can imagine all these things, if it happens, what could be the next progression of this particular kidney cells. So the lead, like the cationic species of some of the other toxic metals, is also capable of acting as a functional mimic of the endogenous ions at a at a intracellular binding sites, and they have also shown that functional substitute for this calcium at the site of this calmodulin protein, which also plays a role in the regulation of intracellular calin addition. The activity of this protein kinase C, which also been affected by the binding of this lead under normal conditions, calcium activates this. You all know, under normal condition, calcium is responsible for activating this calmodulin protein kinase enzyme, right? So this is going to be stopped, which further mediates the numerous intracellular processes and signaling cascades, as you all know, by activating this calmodulin dependent protein kinase C. So if the calcium ions is going to activate this particular enzyme, if the lead and goes and binds to that particular site, functional substitute of this calcium, if the lead is going and binding to that particular going and sitting at that place, so it will deactivate this enzyme, so which further stops this intracellular process as well as signaling mechanisms. You can observe the enzymes will mechanisms will be stopped. Of course, one such example what we have observed in case of if you have remembered long back in metabolism, glycogenolysis and glycogenesis, right? So in turn it is going to stop the activity of your enzyme glycogen phosphorylase, right? So glycogenolysis will not happen, it can be inhibited. So interestingly, it has also been reported like this: the lead is a better activator of this protein kinase C than the calcium ions. And it is also very important to note that action of this intracellular calcium, unlike sorry unlike lead, which are highly regulated. So therefore the binding of this lead to an enzyme protein kinase C, which may activate this enzyme unnecessarily, and it results in deleterious effects, what we have discussed. So this lead can also act as a mimicking agent of the calcium at binding sites of cellular junctions. Many junctional complexes also requires calcium ions in order to maintain their integrity of the membrane, and as well as by binding of this lead instead of calcium may compromise the integrity of the junctional complexes also. In addition to this, exposure to this particular lead also been shown that alteration with respect to the subcellular distribution of calcium ions in the renal cells, and exposure to this lead also resulted in the elevated levels of calcium ions in the cytoplasm and mitochondria, while these calcium ions in the endoplasmic reticulum were depleted. And these changes in the intracellular calcium ratios, which further leads to the significant alterations in the intracellular signaling pathways, as we have all know, we have already come across in signaling, that is in physiology, right, molecular physiology concepts, and eventually it leads to the apoptosis. Interestingly, one thing you can observe here: administration of this calcium channel blockers, Vera sorry verapamil and nimodipine, have been shown to decrease the lipid peroxidation. These are the calcium channel blockers, we can say, and it increases the further activities of the superoxide dismutase and glutathione peroxidase by acting as a blocker. So it will decrease the lipid peroxidation mechanism, and also it increases the activities of the super peroxide dismutase and as well as glutathione peroxidase. And what is the chronic kidney disease and the effects of this lead on the exposure of this lead on the kidney, that is leads to chronic kidney disease, right? Once it was thought that the exposure of this lead was not directly associated with this particular renal disease, but it appears to be it is in a direct relationship between the exposure to the lead and as well as the development of this particular kidney disease, what we have seen so far. So, a study of this industrial workers on this particular lead exposure occupationally, occupational exposure to the lead did not detect over signs of renal dysfunctions, but they have done analysis of this biochemical markers in case of urine, which has been suggested that lead induces changes at the cellular level even if the renal function is not compromised. It can induce the changes at the cellular level even if the renal function is not compromised. So, surprisingly, not surprisingly, we can say that. So, individuals with higher blood lead concentrations, which have higher risk of renal injury, of course, we all know that. A cross sectional study of these individuals in Mexico, they have suggested that serum creatinine levels correlates positively with the blood concentration of the lead high levels of the lead which will be present in the blood. This finding, which provided some information and indicated that the exposure to the lead may also leads to a reduction in the glomerular filtration rate, of course. Similarly, a cross-sectional study of this Korean adults also demonstrated that a positive correlation between the blood lead concentration levels and renal dysfunction. So additionally, they have also observed that erythrocyte lead appears to be associated with this renal injury. So of course we can say that this chronic kidney disease also associated with the high level concentration of the lead which has been exposed to the human beings in the kidney, which will be associated with the chronic kidney disease, or it further leads to the renal injury. So this is all about one heavy metal, that is lead. Now in the next slide we will focus on mercury. So that will be the end of the heavy metal topic. Thank you.